Hey Braves! I know you're all checking out my awesome Dante Hall jersey, and you're wondering, like, why does Mr. Clemens dress like a hobo? I'm basically wearing a Chiefs jersey every day until we're either eliminated from the playoffs or we win the Super Bowl. So you'll probably be seeing me in this getup quite a bit in the coming weeks, because I don't think Patrick Mahomes is gonna be losing any games anytime soon. Alright, anyways, that aside, as I promised last week, this is part 2 of Photosynthesis and Cellular Respiration. And this week, I'm going to be getting into the nitty-gritty of those two particular equations or processes. Now, again, I want to, calm everyone down and make sure you don't freak out, because I'm not going to make you or force you to memorize the very complex processes themselves. But I am going to have you kind of try to remember some of the very basic things about it that I think are the most important. So don't worry, you're not going to have to memorize 24 different chemical names and pathways for these equations to occur. So with that, I want to go on ahead and move into a few quick announcements before I kind of break into going through these equations in more detail. First things first, I'm having some issues with the week ahead and people saying that they actually read the document, but it not showing up on the viewer history. Make sure you're logged in to your student account when you view the week ahead document. If you don't, it will not record the fact that you viewed the document. So I think what has happened for the 3 people who have brought this to my attention is that maybe they were on whatever website, and if you haven't noticed, Google remembers your preference for whatever account that you're in. So if you're always in your personal account when you're using your Chromebook, and then you sign in to the one app, Google Classroom, in order to actually get access to stuff, whenever you click on a link, it will oftentimes open that link up in your preferred account setting, whatever that is. So if it's your personal account that you're always using, even if you're logged in as your student account, when you click on something, oftentimes it will open it up in your personal account. It happens to me every single day and it drives me nuts, but it is what it is. So keep in mind that you need to be logged in to your student account so that you can get credit for having read the week ahead. If I can't verify that you actually read it, I can't give you points. That's not fair to everyone else who actually doesn't. Okay? And it kind of defeats the point of me giving you points for it. Here's the deal. No matter what, on Tuesday, that is the cutoff date for the week ahead. If you don't view it by then, you're not getting points, because it doesn't make any sense to go halfway through a plan and then go back and actually read it for the first time. I want you reading that first before you start working on your classwork for the week. So, there's that. January 25th, we are going to start a brand new school schedule. Those of you who are coming twice a week, instead of hanging out in one class, you will be going to your full class schedule, all 7 of your classes and seminar in those 2 days. You'll do 4 at once on like a Tuesday or a Monday, whatever your first day of the week is, and then you'll do the other 3 in seminar after that. Because of this, my morning availability is going to be drastically reduced because I'm going to be helping students on their work in class during those morning hours. So I will most likely be available in the afternoon. So if you have pressing issues, whatever, you can still send me an email in the morning, but just know I'm not going to get to it right away because I'll likely be helping all of the wonderful students who are finally in my classroom. All right, so keep that in mind. Extra credit this week. So I do have 2 extra credit assignments for you. They are both crash course biology videos, and I have them on an Edpuzzle. The reason I have them on an Edpuzzle is A, it kind of helps you break up the video in kind of chunks so you can think about what was important and what you just watched, but also too, it's an easy way for me to give you a grade. So if you want to watch those 2 videos, I have them on the week ahead as a separate thing. If you want to watch both of those, I'll give you extra credit, 10 points apiece. And those videos go into the minutia and in great detail of how the 2 equations we're going over this week really actually work. So I'm obviously not going to make you memorize those processes, but for those of you who are interested in this type of biology, I wanted to have that available for you, and I also wanted to be able to give you some points for actually exploring that if that's something that intrigues you. So those are up there for you this week if you want. This is going to seem like a heavy week. It's going to seem like there's a ton of things to do. But when you get down to how much time you actually have to put in this week for all of the different assignments that I'm giving you, it's less than 2.5 hours. The first two Ed puzzles, the Amoeba Sisters, they're 7 minutes and 7 minutes a piece. So you're looking at 14, 15 minutes to get 2 of the assignments knocked out. It's not that much time. This video obviously is going to be a little longer because I'm going into more detail, but even this one's going to be shorter than 30 minutes. So if you add those 3 together, you don't even hit an hour worth of time, and then that just leaves you your reading assignment and your reflection for your weekly goal left. So you're not getting a ton of work. I know it might feel that way, but I promise you, this is just as easy as of a week as last week was. So, again, I'm trying to assuage your fears and just, bring down the level just a little bit. All right? Okay. Final announcement. So the reading assignment this week, I am going to count as a quiz grade. As always, with all of my Google Form assignments, you can retake that as many times as you wish to get the grade that you desire. Again, that's always because I want you to learn the material, I want you to understand the material, and I'm not worried about whether or not you got it the first time or the second time. I just want it in your head so that you understand it. So that will be a quiz grade this week, and I'm not making that due until Sunday. So if you have a really busy week and you just can't get to it, well, you have the weekend to get it in as well. All right, so that's it for the announcements this week. Now let's go on ahead and start looking into photosynthesis and cellular respiration. What's it all about? Okay, so photosynthesis. To start off, let's go on ahead and just remind ourselves what the photosynthesis equation is. So as you see here, as it pops up on the screen, you have 6 molecules of CO2 and 6 molecules of water. And when you add some light, you get 1 molecule of glucose and 6 molecules of oxygen. And one thing to draw your attention to is that the oxygen is in the form of O2, which is the natural form of oxygen. You never see, I shouldn't say never, but you rarely see a single oxygen molecule floating around because they're really looking for something to latch onto. So the reason I bring that up is if you look down here, at some point we're going to break some water molecules apart and we're going to have single oxygens hanging out. So when you see this 1/2 O2, that is the same thing as just saying O, and I understand why they write it that way, but it can be confusing. So I just wanted to clarify that. And what's going to happen is once you break apart 2 water molecules, there will be 2 O's floating around, and then they will go on ahead and bond together, and you'll have O2 once again. So this is why you actually get oxygen released out here as a waste product eventually. So anyways, that's that. One thing I wanted to mention. So photosynthesis is broken up into 2 different processes. You have a light-dependent reaction, which is over here, and this is what happens inside of the chloroplast inside of the granum, or the thylakoids. Each one of these little discs is a thylakoid, and when you stack them together, you get a granum. Anyway, so the light-dependent reaction is happening inside of there. And the reason these are green is because there's the pigment chlorophyll in there, and chlorophyll is actually what is absorbing the light from the sun to make all of this happen. So that's what's going on this part. The Calvin cycle, on the other hand, is taking place inside of the stroma, and that's kind of like the empty space inside of the chloroplast. And this process is a little bit more complicated, and I'm going to go into it, but anyways, the Calvin cycle basically is what we end up using the CO2 for, and it's where we actually assemble the glucose molecule for this whole process. Now, the thing to mention before I get into the minutiae here is that the light-dependent reaction is focused on this second part of the process of photosynthesis. So what's going on here is we're creating some important molecules in a light-dependent reaction that are needed on this side to assemble the glucose. And some of the waste products from the Calvin cycle will actually be used over here in the light-dependent reaction again. So this whole thing kind of cycles, and the whole point is just to connect CO2s together so that we have a fuel source. So anyways, let's go on ahead and get into the minutiae. On the light-dependent reaction, you guys will recognize this particular diagram here. On the light-dependent reaction, what we're really focused on is moving 1 electron through the membrane of the thylakoid. You can see this is a lipid bilayer here, and these are obviously protein structures, and each one of these things kind of serves a purpose of moving this electron along And as it does that, the movement of the electron allows hydrogen atoms, and hydrogen atoms are just a proton with nothing else on them, they don't even have electrons, it moves them across this membrane. And that's important because these are eventually used over here on this side to go through ATP synthase to create ATP. Basically, it turns ADP into ATP. So moving these protons back and forth is actually kind of an important part of this. So it's not just accidental. Now, that light that I mentioned before, it comes down and it's absorbed by photosystem 2. And that action there is what splits the water molecule into 2 protons, 1 electron, and then 1 oxygen molecule. And this is where the light absorption comes into play. Now, when that electron moves across the membrane and all the protein structures and all the stuff is happening, as I kind of just mentioned, when it finally gets out to the end of its path, it is responsible for using one of these hydrogens that are floating around and using its energy to create the NADPH. So you have an NADP plus molecule, and because you have this new electron here, it gives this thing right here the energy it needs to combine with the hydrogen protons floating around here, and this creates this new molecule here. So it's a lot of science gobbledygook to basically get to this point, or to explain this. But really the main thing that you want to focus on, so here's kind of how it breaks down. I just showed you that. But the thing you want to focus on are the products. What is it that is being made? Well, if you take a look over here, we're getting ATP out of this, we're getting oxygen out of this, and we're getting NADPH. Now, the O2 is just a waste product. So the cell doesn't really want this inside of it, and it kicks it out. So it goes away. That obviously is what we use to breathe with. The 2 things that it was after is ATP and NADPH, okay? So these are necessary for the next part of photosynthesis. So long story short, the light breaks apart a water molecule. The electron that was attached to that moves across these protein structures. This allows protons to go back and forth, and those protons are used through the ATP synthase channel to create ATP. And eventually the electron ends up over here and gives its energy to NADP+, so it can bind with one of these hydrogen protons to create this. And that was the whole process. All right, so let's go ahead and talk about what we're going to use the ATP and the NADPH for. Okay, so step 2, the Calvin cycle. Now, again, this is one of those things that can be fairly intimidating. And like I said, this is fairly intimidating oftentimes for me because it's a lot of things for me to remember. And the process is a lot more complicated than what I'm about to explain because there's certain things that are happening when you add the ATP and the NADPH that I'm not going to get into. So anyways, the important thing to remember is the last step you had ATP generated from the light-dependent reaction, and you had NADPH. And there is an enzyme that helps the Calvin cycle begin. It basically helps put together these CO2 molecules, which is the first thing, or the first step, is combining the CO2 molecules with this enzyme rubisco. And this rubisco here is actually generated from the final step of the Calvin cycle. If you see, there's this RUBP, and there's leftovers here that are eventually turned back into this rubisco and then reused for the process. So it's called a cycle because it just keeps going and going. It creates some products that it needs for other steps in the process. So I'll move on. So what happens is this rubisco combines with these CO2s to create this next molecule, phosphoglycerate. And the phosphoglycerate is then essentially reacted with your ATP from the last step and your NADPH from the last step to eventually get yourself a glucose molecule at the end. Now, you're going to have some waste products. So you can see it comes up with 6 G3P, and 1 G3P is used to create a glucose molecule. Now, the waste products then give you 5 of these left, and when you add another ATP, you eventually get to 3 of these RUBPs, which are used to make the Rubisco, and the whole process continues again. The point of this whole thing, and I'm going to keep it short, is that we are using the products from a light-dependent reaction to add them essentially to the CO2 that the plant takes in, and to basically combine those CO2s until you get a glucose. And remember, glucose is C6H12O6. So that's all that is occurring in this particular step. Now again, I can get more specific, but I'm not going to. I think that's probably a good enough explanation right there, and we'll go on ahead and cap it at that. Okay, so that leads us into cellular respiration. So we just went through photosynthesis, and now we have a glucose molecule. And if I'm a plant, there's several things I can do with that glucose molecule. I can either send it off to be assembled into one of my structures to create something like fiber or starch or something like that, or I can go ahead and send it over to my mitochondria and start using it as energy. So obviously plants are creating that glucose so that they can use it. They're not making it for you. You just get lucky that you get to eat them and steal it from them after they made it. So now with cellular respiration, you can see the equation, again, as a reminder, is just the reverse of the photosynthesis equation. Photosynthesis is adding water and CO2 with light to create glucose and oxygen, and cellular respiration is taking that oxygen and combining it with glucose to get those waste products that photosynthesis used as the reactants. Okay, now cellular respiration is actually 3 steps in order to get there. If you remember, I can just light a match and I'm using this equation and there's no intermediary steps. It's just straight up burning the paper and creating energy, water and CO2. With cellular respiration, we can't set your cells on fire. Like, cells don't want to be burned. So they have to do this in a controlled fashion, and they have to do this in a fashion where that energy that they release is actually usable for the cell. So that's why it's a 3-step process. And the first step is glycolysis. And glycolysis is an important step because its products get pumped into the next step, which is the Krebs cycle. And then its products are important because then they're used in the electron transport chain, and that is the money maker. That's where most of the ATP is generated when you use glucose. So you look at this and it's like glycolysis and the Krebs cycle only give you 2 ATP apiece. Why would the cell bother with those? But it needs what is created there in order to do the big thing, which is the electron transport chain. And you guys have already seen electron transport in the very first step of photosynthesis. It's a very similar process. So it's going to look familiar. Anyways, so this is how we get through cellular respiration. So glycolysis, you can see glycolysis is a somewhat complicated process. The equation for glycolysis, so that we all are aware, is the glucose molecule plus some new stuff. So NAD plus sounds like the NADPH that we were using before, and it actually serves a similar purpose. It's there basically to transport protons and energy for the next step, just like the NADPH and the ATP were. And the same thing here for the ADP molecules. So these things are your reactants, and then what you get at the end of it is 2 pyruvic acids, which are 3 carbon based. So you'll see that we start off with a 6 carbon molecule in glucose. We end up with a 3 carbon molecule in pyruvic acid. And you can see there's 1 carbon, there's 2 carbons, there's the 3 carbons, and that's what the actual molecule looks like. We then end up with 2 ATP, like I mentioned before, which will actually be used in the next step of the process. And then we end up with an NADH. So you can see, look, there's that H, just like with the NADP and NADPH. Well, with this one, it's NAD plus and NADH. So we're grabbing those protons again to be used for something else. Now, I would walk you through all of this, but I really wanted to just show you that this process, we start off with the glucose, we add some ATP, that changes it a little bit. We add some more ATP, that changes it again. And you're sitting there asking yourself, wait, we're using ATP? I thought we were trying to get that out. And it's like, one thing to consider is there's nothing gained unless something's lost. So we actually have to use some energy in order to get energy out. Anyways, point being, we use some different chemical reactions to eventually change this into the final product, which is the pyruvic acid at the end, and the ATP molecules. I'm gonna leave it there because I don't want anybody's minds to melt out of their nostrils. So anyways, the whole point, this cuts the glucose molecule in half into these two things right here, which are going to be used in the next step. It's basically a more usable version of the glucose. Same thing here, we're getting these as products for the next step. And because we used 2 ATPs in the process, we actually only net 2 ATP here, even though 4 are generated. Because we had to use 2, we only technically get 2 out of the overall equation for glycolysis. Okay, moving on. So the Krebs cycle. When we're looking at this diagram in particular, I want to draw your attention to a few things. So the boxes here that are in purple are showing you what the new name is of the molecule that is created in each step here. So you can see the initial pyruvates that we got from the last step of the process in glycolysis, well, they're immediately turned into something new. So there is an enzyme called coenzyme A, and that is combined with the pyruvates from last time to create acetyl-CoA. And sometimes when I'm talking about these, I incidentally use acetyl-CoA and CoA interchangeably. They're not interchangeable. Coenzyme A is what turns pyruvate into the acetyl-CoA. Now, once we get into the Krebs cycle, we start using that acetyl-CoA that's created, and we combine it with oxaloacetate, and this creates citrate. Citrate is also known as citric acid, and that's why this is also known as the citric acid cycle. Citric acid is a 6-carbon molecule, so we just broke glucose down from a 6-carbon to a 3-carbon in pyruvate, and then suddenly we restore it to the same size it was before, but now it's got a different configuration completely. So now it's in a citric acid. Now, what we're going to end up doing in this cycle, and don't worry, I'm not going to go through each step, is we're going to start generating some things that are needed for the electron transport chain, which is next. So we end up with NADH. So you can see right here, all these red boxes are things that are being generated from the Krebs cycle. We're ending up with several molecules of NADH and several molecules of FADH2. And these are very similar to the molecules that we used in photosynthesis during the electron transport chain. So you'll notice they're just off a little bit in terms of the actual name, but they're serving the same purpose. They're bringing along hydrogen protons, and they're bringing along electrons to go and stick in that electron chain, but we'll get into that in a minute. So basically what's happening here is that we're starting off with this citric acid, this citrate. We're subjecting it to things like NAD+, and that tweaks the molecule a little bit, and then we do some more NAD+, and that tweaks the molecule again. And it continues to tweak the molecule until we start getting out all of these NADHs and FADH2s that we wanted. Okay, so that is really our main goal right here, is to create a little bit of ATP, but it's not the main goal. Our main goal is to generate these two things right here, FADH2 and NADH. And if you look, we end up getting 8 NADHs out of the Krebs cycle, and we only get 2 FADH2s. So all of this to say, it's an enzymatic reaction, so it only starts if you have coenzyme A. So it's an enzymatic reaction. You have to run this cycle 2 times, so 2 times for each glucose molecule to get these products over here, okay? And what is released as a waste product? What is it that the cell doesn't want in here? Well, it's the CO2s. So this is why when you breathe out CO2. All of your cells are going through the citric acid cycle, and CO2 is their waste product. So I'm going to leave it there because I can obviously get much more into detail than this, but I don't think it's really too useful. Again, to sum it all up, we're changing those pyruvates. Essentially, we're turning them into citric acid, and then we're tweaking that citric acid molecule until we get the things that we want for the next step, which are these things right here. So that's really the Krebs cycle in a nutshell. All right, so the electron transport chain. I can already hear all of you cheering behind your computer screens because
thankfully we have finally reached the end. Now, as I said before, the electron transport chain is the moneymaker. It is what is responsible for generating the most ATP. This is also the step where we use the products from the previous two steps in order to generate this ATP. Without FADH2 and NADH, we are not able to get such a big bang out of the electron transport chain. So what happens is that the NADH that was generated deposits its hydrogen proton and an electron. If you remember, just like in photosynthesis, the second step of photosynthesis, we had a molecule that would bring over the electron by depositing a hydrogen, and the electron would go through these protein channels, and it would excite the hydrogen protons to move across the barrier. Well, the same thing is happening here. It's identical. We're just using two different molecules to get this started. Now, what happens is these hydrogens that are deposited or left behind by the NAD and the FADH2, well, they are pumped through these protein channels thanks to the electron that was left behind. It gives it the energy to move across. And same thing here with the FADH. It deposits an electron, and that excites the protein channels, and that allows the H to go across the membrane. And you're asking yourself, well, why do we care about protons being out here in the space between membranes? Well, if you look, there's a whole lot more of these H's out here than there are inside of the matrix. And anytime you have a higher concentration across the semipermeable membrane than the other side, okay, I ran out of space there, so this would be a lower concentration in here. Well, the protons, they want to go away from the other protons. They want to have some more space. So this encourages their movement through the channel that we're really interested in, which is the ATP synthase channel. And if you remember from the second step of photosynthesis, ATP synthase is what's responsible for turning ADP into ATP. So we go through all of this complex stuff just to move hydrogen protons across this channel right here so that it can make ATP. That is the main goal behind cellular respiration. So it's all of this complex gobbledygook to essentially move some protons across the right membrane, or the right protein in a membrane. Now, what happens to the electrons? So if you guys recall, the very beginning of the cellular respiration explanation, I mentioned that there was going to be some single oxygens left over from some of those steps. Well, this is where the electron eventually ends up. It drops off here to the O2 and combines with a bunch of the H's that are flowing around here to create water. And that's a waste product that the cell doesn't necessarily want. Now, it'll use some of it, it's going to keep some of it, but this is how we generate water as a waste product of this whole process. So that's kind of like the final step to kind of wrap it all nice and tight up with a bow. All right, so hopefully your brains didn't melt while I was going through this, and I did a good enough job of explaining it on my end that you at least have somewhat of an understanding of how all of this works, and if not, you at least have some respect for those tiny little cells that are doing all of this work inside of your body. So there you go, guys. That is photosynthesis and cellular respiration.